Okay guys, so we are going to carry on looking at the evidence for evolution today, looking at both the comparative anatomy or comparative morphology, as it's sometimes called, and using DNA evidence as well. So basically, comparative morphology uses analysis of similarities and differences in structures to show evidence of evolution. And the reason why this helps show evidence of evolution is due to two key concepts of evolutionary history, and that's that life changes and adapts over time, and that all living things are related. And the more closely related two things are, the more recent their last common ancestor. So using those two key components for evolutionary theory, by analysing body parts of living things and fossil things as well, we can gain evidence of evolution in that way. So the first bit of evidence comes from homologous structures. So that's when an organ or a bone or another structure shows similar forms in animals. The ultimate structure can be similar even if there are superficial similarities. So as an example there, I've got a diagram of the bones in a bat wing. Okay, so you've got a large bone at the top, two smaller bones, the radius and the ulna, that branch out with a gap in between them. Then a small carpal, which is their wrist bone essentially, the metacarpals where they branch out into the hand bones, and the phalanges which branch out into the finger bones, and then you have the wings, the tissue that makes the wings sort of stretching between these bits here. So a bat wing superficially may look really, really different to our arm as humans, but really the ultimate structure is the same. We've got a large bone at the top, the humerus, two smaller bones in the forearm, the radius and the ulna, and there is a gap between them filled with tissue. And then we've got our carpal, so our wrist bone, our metacarpals in our hands, and the phalanges, which are our fingers and thumb. So the ultimate structure is the same, even though on the face of it, a human and a bat's arm may look very, very different, they've still got the same structure. And that provides us evidence for evolution as it indicates a common ancestor for all species with that structure. So a key part of evolution says that as life changes, things descend from common ancestors. So if we go back far enough, every animal with that structure in their arm comes from the same ancestor, which probably branched off from other life a long, long time ago. The next thing we're looking at is analogous structures. So an analogous structure is when species have structures that function or look similar to a structure in another species, but not due to common ancestry. So basically, for analogous structures, they have a similar feature, but it's not due to them inheriting it from the same ancestor. They've gained it due to having a similar lifestyle rather than similar ancestry. So a good example is wings. Wings for flight have evolved 4 times in history. They've evolved in insects, they've evolved in pterosaurs, they've evolved in bats, they've evolved in birds. And they all evolved in those groups after they branched off from each other. So the branch of life that became insects branched off from the one that became vertebrates a long time ago. Within vertebrates, reptiles, birds and mammals all branched off at separate times, and after branching off, wings evolved. So they all have wings not because they've got a common ancestor, but because they've all adapted to similar environments which made having wings useful. So that provides evidence for evolution as it shows organisms adapting and changing to their habitat or lifestyle. So another key part of the evolutionary theory. Next, we've got vestigial structures or vestigial organs, which are parts that are present in an organism but no longer function. So a good example is whales and snakes. Whales and snakes don't have any need for a pelvis because the pelvis is what supports your legs essentially, and neither of them have legs. But they have evolved from things that did have pelvises, so they do still have a small pelvis, but it has been greatly reduced in size. So that's shown that they've obviously evolved from ancestors that did have a pelvis, but also they've adapted over time to change the pelvis in their body and invest less energy in making bones for that, reducing it over time. So that provides evidence on both fronts. It shows that organisms adapt and change over time, but it also indicates what groups they have evolved from over time. If they just come into existence separately, there's no explanation for why they've got that reduced pelvis there. Another good example would be birds. So plenty of birds have wings but are not capable of flight. Think emus, ostriches, kiwis. So again, they're vestigial organs, they still have wings, but they don't have any actual use for them anymore. So they've adapted their lifestyle over time to no longer need the flight. And then finally, we've got embryology. So embryology involves comparing how organisms look at different stages of development. And basically what we see with that is we see that over later in development things begin to look more different, but early in development they look more similar. So if they're staying the same in development for a long period of time, that's indicating they've descended from a similar ancestor, and the changes begin happening later on in their development process. So now we're just moving on quickly to looking at DNA evidence. So DNA evidence which backs up looking at relatedness between species. It can help us indicate if our ideas on how related species are is correct. So the first thing we may want to look at is called molecular homology, which is basically analysing DNA sequences of species. And species that have fewer differences in their DNA are more closely related. If they only branched off or speciated relatively recently, there's less time for mutations and natural selection to operate on them, and therefore their DNA is more similar. So for example, humans and chimpanzees. Remember, chimpanzees are our closest living relative. We did not evolve from chimpanzees, but the ancestor that we did eventually evolve from branched out into the groups that became chimpanzees and humans. So they're our closest living relative. We didn't evolve from them. They're our closest living relative. But anyway, we have about 98.6% of our DNA the same as chimpanzees. So because we only branched out from them relatively recently, that means there hasn't been a huge amount of time for lots and lots of mutations to build up. So 1.4% of DNA being different is still many, many, many bases of DNA. So that's a lot of differences, but compared to the amount of DNA there is, it's only a really small proportion. And then also we can analyse mitochondrial DNA to study evolution. Mitochondrial DNA is entirely maternally inherited. The reason for that is I've got just a basic diagram of an egg and a sperm there. An egg is basically like a regular human cell, but much, much bigger. It's got a nucleus with only half the amount of DNA, and it's way, way bigger to provide plenty of energy as it begins to develop into an embryo. And basically, all the organelles are from the egg. So you can see I've put in the mitochondria and stuff, so they are only from the egg. The sperm, while it does have some mitochondria in the back part of it to enable it to have the energy to swim, the only bit of the sperm that actually fuses with the egg is the nucleus. So no organelles, apart from the nuclear material, is coming from a sperm. So mitochondrial DNA is entirely maternally inherited. So it's useful for studying ancestry in organisms because we're looking at only along the maternal line. It's also useful because it doesn't undergo recombination. So the DNA remains relatively unchanged. And it also has a faster mutation rate than nuclear DNA. So it mutates much quicker than the DNA in our nucleus does. That's really useful for analysing organisms because it enables us to be able to do more for comparing closely related individuals. Closely related individuals, when their mutations are happening a lot slower, they won't have many mutations that are different at all, whereas their mitochondrial DNA, which is mutating much quicker, will have more differences, and therefore we can analyse things that are very closely related as well as distantly related things as well. Anyway, that will do for that lesson, and keep working hard. Thank you.